Good morning, children. Today we will discuss about sexual reproduction in flowering plants, that is chapter 2 in NCERT biology textbook. In this chapter we will discuss about morphology, structure and the process of sexual reproduction in flowering plants. What is a flower? Flower is a modified shoot for sexual reproduction. Shoot means all the parts present above the soil. Flower is a modified shoot for sexual reproduction. Flowers are the objects of aesthetic, ornamental, social, religious and cultural value. Flowers have been used as a symbol for conveying human feelings such as love, affection, happiness, grief, etc. First, we will discuss about the parts of a flower. The stalk of a flower is called pedicel. What is a stalk? Stalk means the attachment or support of a flower to stem. Flower stalk is called pedicel. The floral whorls or flower parts are arranged or situated in the solen part is called thalamus. Okay. Name the floral whorls or the flower parts. First part is called calyx, which is the outermost green part, contains the units are called sepals. It covers the bud in the young stage, so it gives the protection. Second part is called corolla, which is the second part of the present inside the calyx, which contains the units are called petals, which is the colored part. It attracts the insects for pollination. The third part, it is called androecium, which is present inside the corolla, which contains the units are called stamens or microsporophylls, which produce microspores or pollen grains. The fourth part, it is called gynoecium, which contains the units are called carpels or pistils or megasporophylls to produce the megaspore or ovules. Out of these four whorls, calyx, corolla, androecium and gynoecium are the four flower parts. In that, calyx and corolla is known as the accessory whorls and the androecium and gynoecium are known as the essential whorls of a flower. Because androecium and gynoecium are the important units of sexual reproduction. In this chapter, again we are going to discuss about the events in sexual reproduction. Last chapter we discussed about what are the events of sexual reproduction: pre-fertilization, fertilization and post-fertilization. In the pre-fertilization, again two events are there. What are they? Gametogenesis and gamete transfer. Gametogenesis is the formation of male and female gametes. In the flowering plants, androecium and gynoecium are the gametes. So, androecium consists of stamen, which represents the male reproductive organ. Gynoecium represents the female reproductive organ. Now, we will discuss about the male reproductive organ, that is called the stamen. Okay, the structure of the stamen. It consists of two parts, a long and a slender stalk-like structure is called filament. See that one? Long and a slender stalk is called filament, and the terminal bilobed two partitions are called anther. A stamen contains two parts. First one, it is a long structure, filament, and in the tip region it is called anther. The tip of the filament is attached to the thalamus or the petal of the flower. The other part, one side it is attached to the thalamus region or the petal of the flower. The other region it is attached to the anther. The number and length of the stamen are variable in flowers in the different species. If you were collect some of the stamen, each from different flower species, are arranged them on a slide, we can see large variation in size and shape and attachment of the anther. Different flowers have the size and length of the stamen, length, shape, and everything. It is different from species to species. A typical angiosperm anther is bilobed. That means it is two partitions. It is present in the one structure, and each lobe or each partition having two theca. Again two divisions are there. So total 4. So the anther is bilobed and dithecous. The anther is a four-sided or tetragonal structure consisting of 4 microsporangia located at the corners, 2 in each lobe. Okay, see that one? There are 2 bilobed partitions and each lobe having again 2 separate. So, total 4 microsporangia is present. The microsporangia or the pollen chambers develop further and become the pollen sac. They extend longitudinally all through the length of an anther and are packed with pollen grains. Next, we will discuss about the structure of microsporangium. In a transverse section TS of a typical microsporangium appears near circular in outline. Almost it is circular in shape. Okay. Each microsporangium has a sporogenous tissue surrounded by an anther wall made up of 4 wall layers. Okay, see that one? This is made up with the anther layers contains, there are 4 layers are present. These wall layers are produced from a primary cell, one cell derived from the mother cell. The 4 layers, we should discuss about what are the 4 layers. See that the first layer, it is called epidermis, the outermost layer. It is flattened, only one layer which provides protection. First outermost layer is the single layer. It is giving to the protection of the microsporangium. The second layer, it is called endothecium. Second layer is endothecium because theca is present, thecium, endothecium. It is present below the epidermis. It is again single layer of radially elongated cells with fibrous thickenings made up with fibrous. These cells are hygroscopic, water absorbing. When they lose water, they contract and cause dehiscence of anther for the release of pollen grains. That is the giving to the protection. Again, endothecium cells. Then next, the third layer of cells are called middle layer of cells. These are 2 to 3 layers of cells present between endothecium and the last layer is the tapetum. They store food materials. Tapetum is the innermost layer of the anther wall containing the cells with rich cytoplasm and nuclei it's present inside. And the tapetum nourishes the developing pollen grains. It giving to the nourishes it to the developing pollen grains. So these are the 4 main parts are present, wall layers are present. First one is epidermis, single layer, endothecium, again single layer. Middle layers of the 2 to 3 layers it is present. Inside the middle layer contains the tapetum. Inside that nucleus is present. And it is a tapetum giving to the nourishes the developing the pollen grains, developing pollen grains. It contains the cytoplasm and also sometimes more than one nucleus is present. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. In the center it is present. See that one, the diagram, it is showing that all the parts. First one, epidermis, second endothecium, then the middle layers, inside it is the tapetum. Inside the tapetum, the pollen grains are present. See that one. Then the central tissue is called sporogenous tissue. Next, the microsporogenesis, how that microspores are forming. As the anther develops, anther is growing. The cells of the sporogenous tissue undergoes meiotic divisions to form microspores. 4 microspores are forming. Microspore tetrads are forming. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad. Each one is a potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell through the meiosis is called microsporogenesis. What is microsporogenesis? The process of formation of microspores from the pollen mother cell through the meiosis is called microsporogenesis. The microspores, they are formed or arranged in a cluster of 4 cells, that is called microspore tetrad. As the anther matures, this anther becoming mature and dehydrate, which tissue? Wall layer, that is the second tissue, endothecium. The microspores dissociate from each other, separate from each other and develop into pollen grains. See that one? How the pollen grains are coming out. Inside each microsporangium, Several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. The pollen grains are very small, powder-like structure, and they are releasing each microspore, releasing thousands of microspores or the pollen grains which are released the anthers once it become matures.